Thank you. I'm Jay Olson from Oppenheimer, and it's a pleasure to introduce Dr. David Kendall. He's the Chief Medical Officer of one of the leading innovators in the treatment of obesity based in Denmark, and that is Zealand Pharma. We're also fortunate in that he has a vast array of experience in the treatment of obesity, going back to his pioneering work on the first FDA-approved GLP-1. That was exenatide, for those of you that remember that. And he's also spent a number of years at Eli Lilly. So welcome, David. Thank you so much for joining us here.
Thanks, Jay. Pleasure to see many of you again, and thanks for the introduction.
Absolutely. And thanks again to Leonard Sachs and his phenomenal team for putting together today's program. It's been a great pleasure hearing all the lively discussion around obesity. And I think Zealand Pharma is especially well positioned. You've got 3 molecules advancing through clinical development that I thought we could talk about here today, starting with petrelintide, the amylin analog, and you've also got dapiglutide, the GLP-1/2 dual receptor agonist, and then survodutide, your GLP-1 glucagon dual receptor agonist. So maybe just to start with petrelintide. Congrats on the data that you had at Obesity Week. Maybe if you want to kick us off with just recapping that data and maybe some comments on your phase 2b ZUPREME-1 study.
Yeah, happy to, Jay, and thanks. As many of you may know, we released top-line data in late June on the phase 1b, which was the 16-week treatment interval for petrelintide in 3 different cohorts, what we call mid, high, and high dose therapy, which followed on a previously disclosed data set presented at the American Diabetes Association meeting with the lower dose form. And these were all weekly administrations, long-acting molecule with a half-life of about 10 days, so very consistent and in our hands, reproducible PK. In that data set, the 4.8 milligram exceeded 5% body weight loss after 16 weeks, and the 2 higher doses, 4.8 and the 9 milligram dose achieved weight reductions of roughly 8.5%, slightly greater than 8.5%. Many who saw those data have asked, do we really see dose separation or clarity that the higher dose may in fact achieve greater separation? Remember, these are small data sets, phase 1b, only 12 actively treated subjects. And given the dose escalation scheme, only 6 and 8 weeks at the top doses. So we in the phase 2b are still exploring that top end of the dose range to see if we can achieve greater weight reduction. The other thing that many of you may know is it was a relatively leaner, and to call a group with a BMI of 29 lean shows you the world we live in, but leaner populations who tend to lose a lesser percentage proportion of body weight, even in short duration studies. And they were predominantly male. Males have less inherent adiposity, and in general lose 2 to 3, maybe up to 4% less body weight in longer term treatment studies. So to follow on, we have initiated and are randomizing patients into so-called ZUPREME-1, starts with a Z, which is the first of these phase 2b studies with a primary at 28 weeks and then extending out beyond 40 weeks of total treatment. That will be a full dose ranging study to evaluate efficacy and safety of those doses to take us into phase 3. The 28-week data, for those of you on the analyst or investment side, it will be blinded from even me, so please don't ask, or if you do, I'll give you the same answer. So we'll have a blinded team that is separated from the main clinical team, but that will help us in discussions with the FDA to move quickly to phase 3, which we hope to initiate as early in 26 as feasible. ZUPREME-2, which many of you don't know about, is actually a similar study, relatively shorter duration, but looking at the diabetes population. As many of you know, GLP-1 based therapies tend to have a diminution in the weight loss effect. So for example, not the full-term study, but the higher dose tirzepatide treatment in a type 2 diabetes population achieved less than 12% weight reduction as compared to greater than 20% in the non-diabetes population. It is our belief through a number of mechanisms that the amylin-induced weight reduction, amylin agonist-induced weight reduction will be preserved in the setting of diabetes. So if diabetes is well controlled and there is no need for additional glucose lowering, we see amylin agonism and other mechanisms as potentially very attractive. So that will be part of our phase 2 and ultimately phase 3 program as well.
Excellent. Thank you so much for that comprehensive overview. One of the key observations, in our opinion, based on the 16-week data, was the favorable tolerability profile of petrelintide. And since you have 5 cohorts in your phase 2b study, what's your expectation on the safety and tolerability profile with a more optimized titration schedule in a longer study?
Thanks, and I didn't mention that, but yes, it has been for us a very consistent observation that amylin agonism, even the recent CagriSema data where you saw the success at getting to the highest treatment dose on Cagri alone was in excess of 80%. But we have seen, I will call it very low rates of GI tolerability issues, primarily nausea. The most commonly reported GI adverse event is actually decreased appetite, which is reported as an adverse event, but obviously desirable in this setting. And that only a single patient at the highest dose cohort who actually initiated a slightly higher initial dose, the 1 milligram dose, was the only one experiencing nausea and vomiting resulting in treatment discontinuation. The rates of vomiting have been quite low across the phase 1 studies. And if one goes back historically, I am old enough or experienced enough, as I said this morning, to have been part of the pramlintide for obesity program at Amylin Pharmaceuticals. As you get into type 2 diabetes, in particular in the obese population, the tolerability of amylin agonists tends to improve substantially, something we don't see with GLP-1 agonists, probably because those are populations that are amylin replete as opposed to type 1 diabetes where they lack endogenous amylin secretion. So the proof of principle from pramlintide, and I think the cagrilintide data have supported this as well, is that the GI tolerability profile and then a slower will go with monthly dose escalation in phase 2 and beyond, because this is not a sprint, but rather to further improve tolerability, which we already see is quite appealing, potentially beneficial, can and will differentiate this from the tolerability issues with all of the incretin-based therapies, including our own with survodutide and dapiglutide.
Excellent. And then I just want to go back to something you mentioned earlier about the dose response between the 4.8 and 9 milligram dose cohorts. You indicated that there was a similar degree of weight loss. Do you expect to see a more pronounced dose response in the longer study? And then since petrelintide is so well tolerated, can you go higher than 9 milligrams?
Yeah, so the answer to the latter is no. We don't have exposure data or the tox coverage beyond 9 milligrams. Important to note, and people have asked, how does this compare to cagrilintide in terms of dose exposure? Both are balanced calcitonin and amylin receptor agonists, which we believe is critically important. But we believe both based on that and bioavailability in milligram dose, we'll probably get to exposures of 4, maybe up to 6 plus times greater than what you achieve with the currently to be marketed dose of cagrilintide, the 2.4 milligram. The detail in the phase 1b study that is subtle, but I think important, at least for us who are trying to understand the dose response, is in the 4.8 milligram dose group, Jay. They lost a substantial amount of weight in the first 2 to 4 weeks when they were on the same dose as the or the same escalation scheme as the lowest mid dose in that trial, and probably a consequence of having only 12 individuals with inter-individual variation. That actually set them apart. Thereafter, they lost weight no more rapidly and perhaps less rapidly than the 9 milligram dose group, which gives us at least some indication that the higher end dose range may go beyond the 4.85 milligram dose. The other piece that we've emphasized, as I said, we had a very small minority of females in this population, only 1 to 2 in each of the treatment groups. But the females that we have seen appeared to lose the greatest amounts of weight as we would have hypothesized. So we will balance that. We will stratify for BMI, and I hope early recruitment activities support that effort in phase 2.
Okay, great. And then another potential advantage of amylin analogs is in terms of the lean muscle preservation compared to incretin-based therapies. Can you maybe talk about the importance of this for your petrelintide program and anything that you might expect to see along the lines of lean muscle preservation in your phase 2B study?
Yeah, and I mentioned it this morning. I think the lean mass loss that occurs with rapid weight loss, particularly in the GLP-1 based therapies, is not to be discounted. And even though the FDA says it may not have implications, we are looking carefully at that. Amylin agonists, and this is true of Pram, Cagri, and in our hands, petrelintide, at least in animal models, appears to be innately lean mass preserving. The mechanisms for this may in part be related to its capacity to sensitize to the leptin or adiposity signal. So if the body continues to see fat mass, it will continue to burn fat mass. For those of you who've tried to lose weight through other mechanisms, the body has really effective defense mechanisms, both drops, rapid declines in circulating leptin, and so-called leptin resistance, and this was demonstrated by Alan Baer and Jonathan Roth and others with pramlintide way back. There is an innate sensitization in the hypothalamus to leptin signal with amylin agonism, which may in fact contribute to that preservation of lean mass. In our animal studies, it appears to be very specific for fat mass loss. That doesn't mean you will not lose some lean mass, but particularly in sarcopenic populations, those with decreased strength and or a risk of developing sarcopenia, we think this could be a very important approach, and does not require, for example, an activin inhibitor or a myostatin-based therapy to be re-added, but will, at least in my clinical opinion, keeping the muscle you have is likely better than adding muscle once you've lost it.
Sounds good to me. And then I guess since it came up earlier today, maybe any comments you can make on the new FDA guidelines for obesity drug developers and what impact that may have on your future development plans for petrelintide?
Yeah, I mean, we always sit around and wait for these guidances and, think we'll be awed and amazed by the FDA's insight. I did not see, and others may disagree, huge changes in the guidance. And Philip just nodded his head, so I feel affirmed. Thanks, Philip. That, maybe a bit more specificity around cardiovascular safety that's been part of the EMA requirements ongoing. So from our point of view, doesn't change a lot in terms of the development program. I think the focus on lean or functional muscle mass, using 6-minute walk, grip strength, things like that, will undoubtedly be important to include in future programs, and we have those in place, obviously, for the petrelintide program. The other is subpopulations and making sure it's representative. And obviously for us, including diabetes and non-diabetes populations will be critical, because 20 to 30% of those who will be treated either have glucose intolerance or diabetes. And that's where we think the preserving of the weight loss effect with amylin agonists can play an additional role, but obviously including that, important subgroups that are affected differentially by visceral adiposity or generalized adiposity will be important, and that seems to be reiterated in the guidance. If somebody reads the guidance profoundly differently than me, wrestle me after this, and we'll talk about it.
All right, another topic that we've heard a lot about during the course of today's discussion is the foundational nature of GLP-1 based therapies, forming the backbone of future combinations. Can you talk about where petrelintide fits into the treatment landscape of obesity management? And can an amylin analog replace GLP-1 as a foundational therapy?
Um, if my boss is here, yes, of course it can. But we believe that amylins can serve as a foundational therapy. For those of you who read the commentaries and read the article about GLP-1s and the Boston-based group that had one of the first corners on the GLP-1 space, when I think it was Pfizer who said nothing's going to replace insulin. This was in 1986 or 1987. Oops. I think, for the reasons we talk about, particularly one we referred to this morning, which is if a third of patients are off their GLP-1 based therapy within a month or two, there is a huge population that needs a new foundational therapy or foundational therapies. I remember when I had to beg for ACE inhibitors because, of course, previous generation antihypertensives were preferred because they were free, basically cheap. But now, ACE inhibitors, beta blockers, and the dihydropyridine calcium channel blockers have shown their foundational place. I think I'm not so naive as to think the world will rapidly abandon GLP-1 therapies in 2029, 2030. But I do think that 30% and maybe up to 70% who choose not to take GLP-1 based therapies will require additional foundational, and I'll say therapies, not just amylin based therapies. But clearly at this point, the proof of principle, and I think the clinical data support amylin is perhaps the next to emerge. That's not to say that combinations won't be important, but something you can take, dose escalate, and tolerate. Because all we really see now is sema and tirzepatide. The world looks, as I said this morning, like it's GLP-1 deficient, so they all need it, and they are incredibly effective and have really triggered all these conversations. So I won't supplant them, but I think better tolerated, more easily dose escalated, particularly in the primary care setting where dose fidgeting is an increased burden. Having something that is simpler and better tolerated. I describe GLP-1 based therapies as acceptable, not necessarily well tolerated, because that's all you have to accept.
All right, I'm glad you mentioned combinations. Are there any plans currently underway to combine petrelintide with other mechanisms? And maybe a related question, did you learn anything from the CAGRA-SEMA top line? And is there anything you'll look for in the detailed CAGRA-SEMA results that will help inform any decisions that you plan to make about combinations with petrelintide?
Yeah. So the answer to the combinations is yes. Again, in a GLP-1 dominated space, we can see where addition of an amylin agonist to whatever dose of a GLP-1 based therapy is tolerated, or in the future, you add a GLP-1 based therapy at the minimally tolerated or the best tolerated dose to a maximally tolerated alternative therapy, say an amylin agonist. For those who don't know, petrelintide was developed and formulated to be co-administratable and co-formulatable, and we've done work in the preclinical setting on both, with tirzepatide, with our own dapiglutide, with semaglutide, that show co-administration and or co-formulation preserve the efficacy of both. So we have a compound that's formulated in aqueous phase and at a physiologic pH. I would be naive and probably speaking against our own best interest to say that dual agonist or dual mechanisms, because dapiglutide, GLP-1, GLP-2 is two mechanisms, the survodutide molecule, GLP-1 glucagon. And I think those second signals will, like combining antihypertensives again, will be targeted to specific adiposity-related conditions. So if you have MASH, is a GLP-1 glucagon or a very potent GLP-1 GIP the best approach, or will what we hypothesize with GLP-2, the anti-inflammatory signal that comes with GLP-2, not just around restoring or preserving and improving the gut barrier, but reducing systemic and or tissue-specific inflammation. For those of you who aren't aware, there's some preliminary evidence that inflammation in the CNS is part of obesity-related neurodegenerative disease risk. Glial cells may express or be responsive to GLP-2. Stellate cells in the liver, similarly, it's hypothesized that that can further augment the anti-inflammatory effect, where GLP-1 and other weight loss therapies may reduce the insult from, say, fatty influx or free fatty acid trafficking to the liver. If you can similarly target the anti-inflammatory component, there's a value add. So combinations, yes. Dual agonism, absolutely. Or we'd be spending a lot of time on development programs. We shouldn't be.
All right. I want to make sure we have time to talk about some of the other candidates you have in development, moving on to dapiglutide. There aren't a lot of companies working on GLP-2. Can you just walk us through the rationale and potential benefits of hitting both GLP-1 and GLP-2 with the same molecule?
Yeah, so if you don't know, Zealand has a pure GLP-2 agonist in glepaglutide. Unfortunately, had a CRL returned based on some of the clinical findings, even though it's a rare disease asset for short bowel. So we do have a fairly good understanding of this and are working with external scientists to understand some of those things I alluded to. What are the effects on gut microbiome, gut integrity, inflammatory signals that may be GLP-2 responsive, even though they're not GLP-2 specific, in the liver, in the neural tissues, particularly the CNS, and the inflammation associated with cardiovascular risk. So that asset is a biased GLP-1 dual agonist, meaning the predominant effect is GLP-1. We've seen that based both on clinical response and weight loss in the phase 1 trial, as well as the GI tolerability issues. So it carries all of the things I alluded to in terms of tolerability issues. But we believe that second hit, that second signal, may have a unique place in certain, I won't call them niches, but certain subpopulations with significant risk of adiposity-based chronic diseases.
Okay. And since GLP-2 seems to play a larger role in inflammation, is there any evidence suggesting a stronger anti-inflammatory role of dapiglutide versus GLP-1s?
At least non-clinical data that would suggest it augments some of the inherent anti-inflammatory effects that come from pure weight loss or reduction in fat mass. So we believe yes, and again, much of this is in animal models. We're looking for human correlates that support this. So embarking upon phase 2 programs potentially in metabolic disease-associated steatohepatitis, neurodegenerative diseases, cardiovascular disease. And for those who don't know, even in inflammatory bowel disease, about 30% of those with Crohn's colitis are obese, and sort of shocking that a disease that affects the gut so profoundly would be associated with obesity, but we see a potential value add even in that population.
Okay. I apologize, we're almost out of time, but I don't want to neglect survodutide, partner with Boehringer Ingelheim. Anything you'd like to share with us in the last few seconds here?
Yeah, quickly, we have about 20 seconds, but the second signal, the glucagon agonism, again, this is a bias to GLP-1 receptor, about 7 or 8 to 1 GLP-1 to glucagon, but just a hint, if you will, of glucagon agonism, and I started my ill-fated basic science postdoc looking at glucagon, both its clinical effects and its expression, but it has potential effects on energy expenditure. More importantly, as was alluded to in several of the sessions, it has very important effects on free fatty acid trafficking and metabolism of the fat that shows up in the liver. So if you can further remove the insult of free fatty acid, I would say, beyond tirzepatide and survodutide, the survodutide data on reduction in fibrosis and progression of MASH and even the potential regression in MASH suggests that glucagon hit may have a very important role to play particularly in liver disease and potentially other diseases that are associated with abnormal free fatty acid flux.
All right. Thank you so much, David. We've covered a lot. We'll wrap things up there. It's been a pleasure chatting with you.
Thanks very much, Jay. Thanks, everyone.